Hi, so I'm going to go through some of the key ideas about movement of, or the transport of materials in cells. Okay, so the easiest one, the easiest method of transport that we can think about, the easiest way that things will spread is by diffusion. Okay, so we will have done probably demonstration in class where you've sprayed an air freshener or perfume, or if someone does a really stinky fart, okay, we know that those smells will spread around the room. Okay, so diffusion is the movement of particles moving from where there's a lot of them to where there's less of them. So if I spray perfume in the corner of a room, there's a lot of that perfume concentrated in one area, and it will spread out into areas of lower concentration until it's evenly spread. Okay, and it can do that because it can do it in gases and liquids, because the particles in gases and liquids are able to move. So if you look in the diagram, if you dropped food colouring into some water, you've got a very, very high amount of colour where you've dropped it. The water particles would bump into it, okay, and then it would start to spread around. Eventually, it would be evenly spread. The particles haven't stopped moving, but because they're moving the same amount in each direction, it looks like it's an even distribution of the particles. Okay, so that is quite probably the most common form of transport of materials, or the spread of chemicals from high concentration to low concentration. Okay, now in terms of cells, hopefully you should be quite comfortable in terms of recognising the two pictures and these names. Okay, now I want to concentrate really on the cell membrane and the cytoplasm. Okay, now the cytoplasm is quite watery jelly, and it contains lots of chemicals, and it's where all sorts of chemical reactions happen. Okay, so it might be that oxygen or glucose has to get into my cell, and it will do that by diffusion. So it will do that by this process here. Now it will pass through the cell surface membrane. So the cell surface membrane is what we call partially permeable. Okay, so permeable means that it can pass through. Partially means that there's a level of control to it. So my cell controls, or any cell controls, what can enter and what can leave the cell. Okay, so diffusion will happen of all sorts of compounds, such as oxygen, glucose, carbon dioxide, in both plants and animals, and they will pass through this surface membrane because it's got tiny, tiny holes in it. Okay, but it has a degree of control. Now, what we need to think about is the actual spread of water specifically. Okay, now I've used the word concentration up here in terms of high concentration. Now in this sort of sense, we're talking about the things that are dissolved in the solution. So the concentration in these cases would be those red hexagons. Okay, so it could be sugar, it could be salt, it could be oxygen, it could be glucose, it could be any different chemical that's dissolved in the water. The little blue dots represent water. So if it's pure water, it's all blue, there's nothing dissolved in it. If you've got a dilute solution, then you've got a few of the red ones but lots of water, and then in a concentrated solution you haven't got very much water at all, you've got more of your chemical. Now we need to think about how the water moves into and out of cells, okay? And that's a process called osmosis. Now I'm going to use a different term to avoid confusion, okay? So when we're talking about osmosis, we're talking about the diffusion, the spread of water molecules, okay? But we use the term water potential instead of concentration so that we don't get muddled up between those two words. So osmosis is the diffusion of water molecules from a high water potential to a low water potential across a partially permeable membrane. So what that's saying is that water moves from where there's a lot of it. So pure water has a high water potential, and it will move into a solution with a low water potential. So where there's not very much water. So it'll move from where there's lots of water to where there's less water. And this partially permeable membrane is my cell membrane. Okay. Now, in most cases, if water is available, then it will move quite freely. Now, like I said, the cytoplasm has got things dissolved in it. Okay, so for example, we could think about it as being a dilute solution. Okay, so if I've got water outside my cell, then the water is going to try and move into it. Okay, so some of these water particles are going to try and move into it. Now, if I surround a cell with pure water, let me go this way. What's going to happen is that lots of water is going to enter the cell by osmosis. So you can see there that my cell, this is when, at this point here, that the cytoplasm and the surrounding solution have got the same concentration. So there's nothing, they're both the same. So we've got a dilute solution inside, a dilute solution outside. So the water doesn't need to move. If the water is moving into the cell, so if it becomes more concentrated, or I put it into a pure water, it will start to swell. Okay, so it's got bigger, it's changed shape and size. Okay, so it's actually pushing out now against the cell wall. Now, because plant cells have got a cell wall, that gives them support and gives them extra strength, so they can fill up with water and it gets really tight and sort of is pushing against the wall, but it won't burst. And we call that turgid when it's very, very full. And that cell wall is giving it the support. Now, if I put my cell into a concentrated solution, so I'm surrounding it with a higher concentration, then the water will try and move out of my cell. Okay, so what will happen is that the water will move out of the cell and my cell will start to shrivel up. Okay, so the inside part here, okay, has actually started to shrink. If it gets so bad, if it shrinks so much that it pulls away from the cell wall, then we say it's plasmolysed, okay? And when you get to this point, there's no going back. If you've got a plant at home and you forget to water it, okay, if you forget to water it, what will happen is that the cells will start to shrink, and then if you water it, it'll go back to normal, and then if you forget, it'll start to shrink again, and it wilts and it falls over. And if you don't let it wilt too much, then it can go back to normal. But once it gets very low and it's plasmolysed, then there's no going back. Okay, so osmosis has an impact on plant cells. It also has an impact on animal cells. Okay, so again, at the moment, this is what a red blood cell looks like. The concentration is the same inside and outside, so the cell looks absolutely fine. If I put it into pure water, then what would happen is that the water would enter my, sorry, I'm just going to have to restart that. The water would enter my cell and it would start to swell up. Okay, so you can see that my red blood cell is starting to swell up. Now, because an animal cell only has a cell membrane, eventually what will happen is the red blood cell will burst. Okay, so the cell membrane isn't strong enough and the cell will burst. If I go the other way and I put this into a concentrated solution, then water will leave my red blood cells and it will shrivel up, okay, in just the same way as the plant cell did. So in that respect, they're very similar, but when you go the other way, it's just that slightly bit different, okay? So this is it being absolutely normal. When I put it into pure water, if I put it into a concentrated solution, then the water will leave by osmosis, okay, and it will shrivel up. Now, those two ideas are the basis for the required practical, okay? So water will move into and out of cells. Now, this example has been done using eggs. So they've digested the shell in vinegar, and you get this sort of rubbery egg left. So it's the membrane that's surrounding the egg. Now, in this case, this was the size of the egg to begin with. If the egg has got bigger, okay, so if it's grown in size or if it's got heavier, that means that water has entered it by osmosis. Okay, so they can do this experiment with potatoes, you can do it with gummy bears, you can do it with carrots, you can do it with cells, you can do it with all sorts of different things. But if it has got bigger or the mass has increased, the reason is that water has entered it by osmosis. Okay, now in this case, you can see it shriveled up a little bit. So in this case, it's got smaller, it's lost mass because the water has left by osmosis. Now those are the really, really basic ideas that you need to remember in terms of any exam question about osmosis. If it gets bigger, so it could get longer, it could get, you could say bigger, if it's just a picture where you can see a change in size, it could be a change in mass that it's increased, right? If it gets bigger, then water has gone into it by osmosis. If it's got smaller, then water has left by osmosis. Okay, now this is the sort of picture that you might be given. So in this case, I've got a similar sort of a bag, like a cell simulation. Because it's got bigger, I know that water has entered the bag by osmosis, okay? And I can look at that because it's dilute out here, so there's lots of water and it's concentrated inside. So that's the reason why the water is trying to even itself out by moving into the bag.